I think in the last week's class you might have got what do you mean by cytoplasm, what do you mean by cytosol, what do you mean by what is present inside the cell organelles. Um, so this class we will discuss about two cell organelles. The cell organelle means organelle means little organ. Why they have given this name? Because these have a very specific these structures have very specific functions, and they play its own role inside a cell. Because of this reason only they have given called these structures are cell organelles. So the first cell organelle, and which is one of the most important part, which is called as the nucleus. You can see the diagram of nucleus here. It is a 3D diagram, which is a 3D diagram. You can see here. Nucleus, it is a double membrane structure, and it is present to the middle of the cell, not exactly middle, somewhere middle of the cell. And nucleus is considered to be the brain of one particular cell. Why it is considered as the brain of a particular cell? Because it carries some genetic materials inside it. Since it carries some genetic material, and genetic material like DNA, it controls everything. That is why it is called as the brain or control center. So this nucleus is having many pores inside that, and the first record of this nucleus was given by Robert Brown. That because the nucleus is first discovered by Robert Brown, and when we look this nucleus under the microscope, it will be very darkly stained inside in a cell. So why it is darkly stained? Because it is having many condensed areas of the genetic material. We will come to that later, how they are condensed and what they are. We'll study that later. Because of that, it is very dark structure when we see under the nucleus, and inside this nucleus we will be having the genetic material. I think you are understood what is the structure of this nucleus. And when we comes here, when you see inside this nucleus, you can see like this. This is electron microscopic structure. The outer, that violet color area, is the nuclear envelope, and you can see nucleolus chromatin structures like that. So whenever we are studying about this genetic material, that is DNA is present inside the nucleus in a thread like structure, which is called as chromatin. And we say that it is present inside chromosome, and as a structure of chromosome we can see this genetic material DNA in the same of chromosome only when the cell is dividing. Almost all the other times, whenever the cell is not dividing, on almost all the time, this will be inside the cell like a thread like structure, which is called as chromatin. And when you see inside this, again you can see that there is a very condensed area which is called as nucleolus. This area is responsible for the RNA biosynthesis. That means they are responsible for synthesizing RNA, which we'll study later. And the first time the nucleolus, this structure, was discovered by a person known as M. Fendona. This is he is a person who, Italian scientist, who found out the nucleolus. And I think you got this. And the nucleus is to be the exactly center. There are many pores are there. You can see many pores on the surface, and if you see here also, you can see that many pores are there. These pores are very important because signals has to come in, come out. There are many movements has to takes place, so these pores should be very very important to that. And the next structure that is attached to this nucleus, very much attached to the nucleus, is known as the endoplasmic reticulum. And you can see this twist and turn structure that is known as endoplasmic reticulum. Endo means inside, plasmic means inside, it is in a plasma stage. Reticulum means it is found in a network like structure. That is why it is called as endoplasmic reticulum. And this endoplasmic reticulum is very important to a particular cell, because this endoplasmic reticulum is the area where proteins are synthesized. I will make it clear. So whenever you see this twist and turn structure on this, you can see some bead like things are present, small bead like things, and they have represent there it as ribosomes. Ribosomes. If ribosomes are present, that particular endoplasmic reticulum is called as a rough endoplasmic reticulum. And if the ribosomes are absent, that means if there is no ribosome, they are called as smooth endoplasmic reticulum. You can make out from the diagram. And there are many folds on that endoplasmic reticulum, which is called as cristae. And why this endoplasmic reticulum is attached to the nucleus? Because ribosomes are the protein machineries. That means if anything has to function inside a cell, protein has to be synthesized. If protein is not synthesized, the cell will never get to know what it has to do. So that message, which protein to synthesize, that particular message is present inside the nucleus in the form of DNA, which will later converted into a protein. And to convert that, the ribosomes are very important. Convert into protein, ribosomes are very important. That is why these ribosomes are attached directly to the endoplasmic reticulum. That means whenever the message comes out through the inside, it will directly enter in. If it is to be protein synthesized, it will directly enter into the ribosome, and they started to synthesize the protein, and the proteins will be given to the packed and given to the necessary areas wherever it is required. That is one of the main function of the endoplasmic reticulum. The second is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, since there is no ribosomes are present inside that, they will never take part in the synthesis of proteins, but they will take part in the synthesis of lipids and fatty acids. So I hope you got this structures what they are. I have given some questions. Kindly do the questions, and if there is any queries, please